My name is Sina Sekheche, and this lesson is brought to you by the Free State Department of Education in collaboration with Pagama Research and Development. Right, so as you are preparing for your paper 2, we are going to be doing our revision for paper 2 today. So let us check the topics for paper 2. So your topics will be weighted as follows. Right, so in term 1, you did DNA code of life, and that weighs about 18%. That is 27 marks of your total paper out of 150. And meiosis was also a topic that you did in term 1, forms part of paper 2, carries 21 marks, that weighs 14% of the paper. Then the topic that was overlapping into 2 terms, that was genetics and inheritance. Just look at the big marks here. 48 marks out of 150. So that is almost 1/3 of the paper. And together with evolution. So these 2 topics here are quite a big topic. So you can spend a lot of time here so you can collect those maximum marks. Then your paper will also be out of 150. This is the structure. And then what are the possible questions that you can expect for DNA code of life? Questions can arise from terminology, interpretation of diagrams. I think you've seen from the previous question papers. Tabulation, you can be asked to tabulate and give a description and calculate. Anything can come out of this topic. Meiosis, the same. Terminology and diagram interpretation, description of a crossing over. You must know what happens during crossing over, all the details. And the tabulation of the differences between meiosis 1 and 2, the differences between meiosis and mitosis. So some of these are some of the skills that you need to have when tackling these topics. Right, for DNA and RNA, the scope or the content that you need to study for DNA and RNA is obviously you need to know the structure, the location, the function, remember. And then DNA replication and its significance, you need to know to remember that. DNA profiling also forms part of that. Then protein synthesis, the 2 stages, that is transcription and translation. You must be able to describe what is happening in those processes. So we're not going to waste more time talking about what you need to do. So we rather just get on with it and get started. So with your DNA and RNA, just take a look at the structures and try to remember the facts that you need to remember about the structure. So we will start with DNA. Your DNA, remember, it is a double helix structure. You're not allowed to use any other word because the exam guideline tells you it is a double helix. Don't lose marks unnecessarily. Then you must remember, so it means you see the 2 strands forming a helix. So it is called a double helix. And the type of sugar that you find in DNA, meaning the D stands for deoxyribose sugar. Remember, if the word is too long, you are allowed to break it into pieces. Sometimes you can break it into 2, sometimes you can break it into 3. So here we have deoxyribose sugar. So you can break it here. Deoxyribose sugar. Then that is the type of sugar that you find in DNA. And this molecule is a very long molecule. And its building blocks, the monomers, remember the monomers are the building blocks. It is made up of nucleotides. So this black dot over here, this pentagon over here, and this shape, yellow shape here, that looks like a cut down rectangle in a shape of a triangle there. It is a nitrogenous base. Then you know you have your phosphate group and your sugar group. So all of these, they make a nucleotide. They are building blocks. So if you go down this molecule, you can see those building blocks. Then coming to RNA, you must remember that the type of sugar that you find in RNA, the R stands for ribose. So the type of sugar that you find in RNA is ribose sugar. And RNA, unlike DNA, it is a single strand. So there is no other strand that is coming from here. If you remember the shape of mRNA and tRNA, then you can just see that it is only just a single strand. Then the length, mostly the length are short. Remember, tRNA can only bring the required amino acid with the matching anticodon, so those are very short molecules. So it is also made out of nucleotides, meaning the building blocks. So let us take a look at the nucleotides at a closer look. This is a structure or a diagram of a nucleotide. So your pentagon here is gonna represent your sugar. So the pentose sugar is just the general name for the types of sugar that you find in nucleic acids. But if you are talking about DNA, obviously the name of the sugar is going to be deoxyribose sugar. And if you are talking about RNA, the name of the sugar is going to be ribose sugar. And then up here, it is represented by the dots. These dots represent the phosphate group. And these, the phosphate group together with the sugar groups, they form the backbone of DNA. So this is the backbone of DNA. And then you have nitrogenous bases. Your nitrogenous bases will be matching, if it is a DNA, with the complementary ones. So what are the names of the nitrogenous bases? We have 4 nitrogenous bases. Remember, the names are guanine, cytosine, adenine, and thymine. However, in RNA, you do not have thymine, but you have uracil instead of thymine. So where you are dealing with RNA, where you see T in the DNA, you know you are going to replace it with uracil, as we do not have thymine in RNA. So you must remember that one. So that is the basic structure of a nucleotide. Another thing that you need to know about the nucleic acids and the DNA is DNA replication. You will be asked this question maybe for 5 marks, 6 marks. How does DNA replicate itself? Remember the steps. There are 3 basic steps that you can get out of 6 marks. So if you are sitting there, you are not able to get 3 out of 6. Please, please, please make sure for tomorrow's exam, you are able to get 3 out of 6. Let me show you what are those 3 that you can collect for free without even knowing the entire process, but you can collect marks with these ones. And remember, you do not leave any question unanswered. All the spaces must be filled. So make sure that you collect as much as possible marks from any question that you are given. Right. The first thing, the double helix unwind. Pay attention to this. We no longer say DNA unwinds. You must tell us that it was a double helix, now it is unwinding. Because someone might not know what the shape was before. So for you to get full marks for unwinds, please say the double helix unwinds. I think everyone will remember this step. And if you did not learn throughout the year a method of learning with your body or with your fingers, please, when you are sitting in the exam room, use your fingers or try to memorize the body things that you were doing to remember this process. So I know some of the teachers can be very creative with this and they can teach you different ways on how to describe this process. So the first step that you must get, this is the first one that you must get no matter what. The double helix unwinds. Then the weak hydrogen bonds between the nitrogenous bases are going to break and the DNA strands or the 2 DNA strands are going to unzip. So all of this is 1 mark. If you say DNA unzip, you're going to get 1 mark. Or if you say the weak hydrogen bonds break, you are still going to get that mark. But you are not going to get 2 marks for writing 1 weak hydrogen bonds break or the DNA unzip. So together it's just 1 mark. Right, so you can write either one of that. So by now you have 2 marks. The double helix unwinds, the weak hydrogen bonds between them breaks. And you know, when something breaks, then it's going to cause what? The unzipping. So hence it is 1 mark for that. And then what happens after that unzipping? Each original strand serves as a template. Now, if you say the 2 things have separated or DNA, the double strand, has unzipped. You must now tell us what happens to each one of them. That is where you are going to get your third mark. So you are going to say each original strand, please use original because we are making copies. Copies are going to be new copies. Remember, even when you go to the police station, you make a copy from your original. So the original ones are going to serve as a template, right? And then that is where you get your next third mark. Please make sure that all these three, at least you obtain that 3 out of 6 marks that you can be asked for this process. So that is 50% for this question, which can be fairly good. Then you can further obtain more marks by saying what? This template is where the complementary strand is going to be built. Then how is it going to be built? That is the next thing. The free nucleotides build a DNA strand onto each of the original 2 strands by, this is the part that is important, attaching to their complementary nitrogenous bases. So remember we said A pairs with T, C pairs with G. So those free floating nucleotides, the A's and the T's and the C's and the G's that are floating here, they are the ones that are going to come and form a complementary pair, making a new copy of DNA. Then this results in 2 identical DNA molecules. Then each molecule consists of 1 original strand and 1 new strand. I think even this one, you can obtain marks here. So even if you've forgotten how does it happen, but at the end, you must tell us that each molecule consists of 1 original and 1 new strand. You are going to get a mark for saying that. So please make sure that you obtain all these marks when you are explaining the process of DNA replication. So a DNA is making copies of itself, remember? Then another process that you need to know, very important with the nucleic acids, protein synthesis. I know most of you want to cram the whole thing without understanding. And please make sure that you follow the processes in terms of diagrams. What is happening? Because you might be given different diagrams, so you must be able to interpret per diagram. Right. So with protein synthesis, this is the process where proteins are manufactured. So how does DNA manufacture proteins? Yes, because it carries the code to manufacture proteins. So we start from scratch. We start in the nucleus with the first process being transcription. So what is transcription? This message needs to be transcribed from DNA to mRNA. So this process happens in the nucleus. What is this process, or how does it happen? You get marks for explaining. Like with DNA replication, you are going to start by saying the double helix unwinds. Although this is a different process, it starts the same because you cannot copy a code if the double helix of the DNA molecule, it is still winded up. So it needs to unwind and open and expose the code that needs to be copied. So that is why it is going to unwind, and the weak hydrogen bonds between them are going to break and the 2 strands unzip. You get a mark for that. So these are the 2 marks or the 3 marks that we've spoken about in DNA replication. You can still obtain those marks here with protein synthesis. Right. Then 1, pay attention here. The difference between protein synthesis and transcription and DNA replication is that here, not 2 original strands, only 1. 1 strand acts as a template for the construction of mRNA. Remember when we were doing DNA replication, we said 2 strands acts as a template. So for transcription, pay attention, it's only 1 strand that is going to act as a template for the construction of mRNA. Then how is mRNA going to be constructed? The free RNA, pay attention also to that. In DNA replication, the nucleotides that were flowing, they were the DNA nucleotides. But here the free floating, remember the nucleotides are just floating around in the nucleus, in the cytoplasm. So you need to be specific to say when you are talking about transcription, you must say the RNA nucleotides. And when you are talking about DNA replication, you must specify that it is DNA nucleotides. So those are the differences that you need to look out for. Now, the free RNA nucleotides from the nucleoplasm form mRNA. So how do they do it? They just come like they did with DNA replication. So they are going to complement that strand of that original strand of DNA. Let's say this was the copied strand. Here it is. So all of these UCA, GCA are complementary to the original strand that formed a template to form the mRNA. Then mRNA complements the DNA in the nucleus. So this is what we were just talking about now. So all this mRNA that is formed, the nitrogenous bases are going to complement what you had in the DNA strand, that one strand that formed a template. Now you have mRNA. What is going to happen now with this mRNA? We are still forming, or we are still following the process of protein synthesis. So because you've formed mRNA, the process is not done yet. Remember, this mRNA needs to further undergo certain processes. So what is going to happen? This mRNA is going to move. You see this arrow here? It's going to move out of the nucleus through the nucleopore. So this is this stage here, number 6. We are now entering the second phase, which is translation. Translation. How do you remember which one is which between transcription and translation? I would say translation sounds like later, and then transcription, you can just remember that this one will be your first one, and then translation comes after transcription because it happens later. You can say it sounds like later, translation, later. Then it happens in the ribosome. What is happening there? The coded mRNA is going to leave the nucleus through the nucleopore. You're going to get a mark for that. And then it is going to attach itself on the ribosome here. You see it has attached itself. This molecule here is your mRNA that was made inside the nucleus. But now it has attached on the ribosome. I hope you know that this shape here represents a ribosome. Right. Then tRNA pick up amino acids. So this tRNA is going to pick up these. The circles or the dots on top of the tRNA are called the amino acids. It's going to pick up these amino acids and complement mRNA in the ribosome, meaning it's going to bring the anticodon that complements the triplets on mRNA. And the triplets on mRNA, remember, we call them codons. So the anticodons on tRNA are going to complement they are going to complement the codons on mRNA to bring the required amino acids. And these amino acids, we are told that they are joined together by a peptide bond. You just tell us now about the bond. The peptide bond to form a protein. Then you know that a protein is formed by a chain of 22 amino acids. So this is the process of how the protein is made. So you can be asked to explain only translation or transcription. So please be on the lookout for such a question. Then another aspect of this topic is DNA profiling. I think you've seen from all the question papers that you've done even this year, now you are able to interpret this DNA profile, because when it is like this, it is a profile. But the method of getting this is called DNA profiling. So what is happening here? This is a technique that is used to identify the sequence of bases. Remember the bases, your adenine, your guanine, your thymine, your cytosine. Those are the bases. So DNA profiling, they are trying to determine or identify the sequences of those. Now, the nucleotides, meaning all the building blocks, are separated. Why do you separate it? Because you want to see the nitrogenous bases are separated from each other into the order that they are found in the strand of DNA. Then they will be put in that order. Then the nucleotides are the ones that are going to appear as dark bands. So you see these dark bands here. You know now they represent what? The nucleotides. And you know now they are arranged in a sequence or in the order in which they are found. And no two people's nucleotides are arranged the same, exactly the same, except for a case with identical twins or genetically identical twins. Then DNA profiling is different from fingerprints. Remember, we no longer use the word fingerprints. You must just say DNA profiling. That is the terminology that is accepted. So go to the exam guideline, check the definition there that you need to state if you are asked to state what is this process or what does this process entail. So you must say it as it appears in your exam guideline. Then some key points to remember that you can also go and cover and use the worksheets that are provided to also cover some of these aspects. So you must also remember that the history is important. Who discovered DNA? Remember the names of those scientists, Watson and Crick and Franklin and Wilkins. Just a brief paragraph to remember what were their roles, who found the X-rays or who took pictures of the DNA, who came up with the double helix structure, and then what are the structural differences between DNA and RNA, the differences between, we've done this also, DNA, mRNA, and tRNA. DNA where you find it, mRNA where you find it, tRNA where you find it. Then they differentiate between DNA replication and protein synthesis. Remember when we were talking about, when we were in replication, we talk about two strands forming a template, but with protein synthesis, only one strand acts as a template to form mRNA. Then transcription and translation, you must learn those off by heart. We've just revised them. DNA profiling, you've just remembered that this is something that you also need to know and apply. Check the questions in the worksheet. They are beautiful questions that are addressing all of this. Then paternity testing involving both parents and the child. So you must also remember that DNA profiling can be used to check for paternity. There are cases that you can identify and remember the uses of DNA profiling. Then the uses and interpretation of DNA profiling. So those are the key points that you need to remember for this module that will put you at a better, for this topic, they will put you at a better position to pass. Then we move on to the next topic. Our next topic is meiosis. Meiosis carries about 21 marks out of your 150 marks. What is it that you need to learn? Remember your DNA replication before meiosis, what happens? Then you must remember the first division of meiosis or the first meiotic division. What happens there is just one diploid cell that gives rise to two haploid cells. Then the second division, remember, you have meiosis 1 and meiosis 2. So in meiosis 2, it is those two cells that are coming here. They are dividing further to form 4 haploid cells that are called gametes. Remember, meiosis, we are forming gametes. Then, but the biological importance of meiosis, crossing over introduces genetic variation. Very important. And this also ensures that sex cells have a haploid number. This is a very, very important function also. Variation, because we need it, as we saw with Charles Darwin. Then also to maintain the chromosome number. Let's say we are talking about the humans at 23 haploid, so that when let's say this is a sperm cell at 23, when it fuses with the ovum of 23, you have an organism of 46 chromosomes. So this number must be maintained at 46. So the function of meiosis is to maintain this, to ensure that sex cells or gametes, remember, sex cells are gametes, have a haploid number. So a haploid number for the humans will be 23. So you must remember all of that. And terminology. With meiosis, like with other topics in life sciences, there's a lot of terminology that comes with it. And for your question paper, in which the structure of the question paper, you are going to find questions on terminology in question number 1.2. Then you must familiarize yourself with terminology such as homologous chromosomes, bivalent chromosomes. What is a chromosome? What is a chromatid? Because sometimes you confuse the two. Then what is a centromere? What is a centrosome? Then what is crossing over? What is a chiasma? What is a karyotype? Nondisjunction, karyokinesis, and cytokinesis. So these are some of the terminology that you need to familiarize yourself with meiosis. Then we start with the phases of meiosis. What happens? Remember, before the phases start, a cell needs to prepare itself. It needs to duplicate its DNA. So this first phase is going to be called interphase. So this is a preparation stage. So what happens during interphase? DNA replication occurs, meaning it is doubling the genetic material. They will ask you, why is it the DNA replication happening? It is doubling the genetic material to be sufficient for cell division. Then each chromosome will now consist of 2 chromatids joined by a centromere. So if it was a single chromosome, now the chromosome is going to be like this and joined together by a centromere. So it means it has doubled its genetic material if it was single, and now it is double. So that has also doubled. It has copied, it has replicated, you see. Then the DNA helps to double the genetic material to be shared equally during cell division. So we want to have sufficient to be shared equally. Then let us address the issue of unreplicated chromosome and replicated chromosome. There's a question that will always come when you are dealing with a meiosis question that will ask you how many chromosomes are at the end of the cell. And some of you will find a cell like this. And then there will be this, and then sometimes this, then this. And you will be tempted to say, this is a chromatid and this is a chromatid. Then when you are asked how many chromosomes are in the cell, you will say 1.5. I know, because in your mind, you are already taking this chromosome and joining it together with this one to make 1. And this one, because it does not have anything to join with, you say it's a half chromosome. Actually, these are full chromosomes, but we say they are unreplicated chromosomes. This is a single chromosome as it appears in a cell before DNA replication takes place. So remember, this is a single strand chromosome. So you say it is unreplicated. Why? Because it has not made a double copy of itself, like in this case, like this one, this X that I've drawn here. So you see, these are full chromosomes, but we say they are single unreplicated chromosome. Then what is a replicated chromosome? A replicated chromosome will be that one that consists of 2 chromatids joined together by a centromere. So let us take a look at the diagram that shows this. So when you take a look at this diagram, the first diagram shows you what we were talking about. Unreplicated chromosome. You see it is a single-stranded chromosome, but it does not have a partner. But you cannot say this is a chromatid when it appears singly. you don't refer to it as a chromatid because a chromatid does not appear singly. So this will be your unreplicated chromosome. Then once you see them joined together like this in this diagram over here, number 2, you see now we can call them chromatids. Why? They are chromatids of a chromosome. This whole thing is a chromosome, but it is made up of one chromatid, two chromatid joined together by a centromere. That is the only time you can refer to this as a chromatid. I hope you remember that. So this will be your replicated chromosome and this will be your unreplicated chromosome. Then in that scenario that we were talking about, so when you have something like this, and then you are asked how many chromosomes, the answer is going to be there are 3 chromosomes, or you can say 3 single-stranded chromosomes or 3 unreplicated chromosomes. But the most important answer is for you to see that there are 3. Right. Then the next phase that you need to be able to identify is prophase, which is where most of the things are happening. Remember, interphase only happens once, and then once it happened, the cell starts dividing up until the gametes are formed. Then those gametes will undergo interphase again, and then cell division will continue in those new formed cells. But for now, let us now go into the next step, the next phase. How do you identify this phase? When you are given a diagram, there are certain things that you need to look out for. You need to look out. If you are talking about prophase 1, number 1, you must see the spindle fiber that is starting to form. You see here on the side. You are going to need the spindle fibers at a later stage. So they start forming where? At prophase 1. So when you see them start forming, you must just remember them. This must be prophase 1 because it happens at prophase 1. Then homologous pairs of chromosomes are visible. So you see chromosomes are in homologous pairs. You have, when we say homologous pair, remember we are referring to paternal and maternal chromosomes. So when you see them attached like that together, you know that this is in prophase 1. Then bivalents are also visible. Then crossing over takes place. How are you going to see crossing over? You will see chromatids of non-sister chromosomes touching or overlapping at a point called chiasma and exchanging genetic material. And once you see the colors flipping, then you know that crossing over is taking place there. Then prophase 2, how do you know now? Okay, that happened in prophase 1, but in prophase 2, what is different? In prophase 2, the nuclear membrane and the nucleolus disintegrate. So once you see that disintegration, then you know that is prophase 2. Remember, prophase 2 is going to happen after the division of the first meiotic division. So this will be the first phase that starts the second meiotic division. Here, there is no crossing over. No crossing over at prophase 2. Crossing over only happens at prophase 1. But at prophase 2, no crossing over. You must just remember that. And here is just a reminder to say crossing over brings or causes variation, which is one thing that you need for the rest of your paper up to evolution. You must know this off by heart. Then the next phase is metaphase. Metaphase. Middle. Metaphase. So in metaphase, whether it is metaphase 1 or metaphase 2, you expect to see things in the middle. Once you see things in the middle, you know it's metaphase. But then you need to identify what are those things that are in the middle. Are they chromosomes or are they chromatids? So let's check. So for metaphase 1, pairs of chromosomes align at the equator. So everything is at the equator, that is at the middle, but the chromosomes are still in pairs. So they align here. So you are going to say this is metaphase 1 because I see the chromosomes or pairs of chromosomes aligned at the equator. Then random arrangement of chromosomes takes place, and this is also another source of variation because those chromosomes are randomly arranged. Remember, you don't say random assortment. The correct terminology there is random arrangement of chromosomes. So these chromosomes do not align according to a particular formula or a particular will. They just randomly arrange themselves. So that brings up genetic variation also in our gametes. Then that is meiosis 1. For meiosis 2, you are going to see them at the middle, but what are you seeing? You are seeing chromosomes. Chromosomes align singly at the equator. They are chromosomes, but now they are single. There is no pair here. You see, there is no pair. Only single chromosomes aligned at the equator. Then you know that this is metaphase 2. Random arrangement of chromosomes also takes place. Then that is your metaphase. Then your next phase, because they are aligned, what must happen? They must be separated or they must be pulled apart. That is your anaphase or your apart phase, anaphase. But you don't say apart phase. So apart is just to remind you that anaphase, things are now pulled apart or to the opposite poles. So anaphase 1 and anaphase 2, how do you identify them? Anaphase 1, you start seeing the spindle fibers contract. Then the chromosomes move to the opposite poles. Remember, the spindle fibers are attached to the centromere. Then, if the spindle fibers fail to separate a pair of chromosomes, then Nondisjunction is going to occur. Do you remember that? And 2 gametes are going to receive an extra copy of the affected chromosome, and that can result in Down syndrome if this happens at chromosome pair number 21. I hope that story is still fresh in your mind. Then in anaphase 2, what do you see? The centromere is going to split. The centromere. Then when the centromere splits, remember, let's follow. What is the centromere? The centromere is this part. Remember, that is joining the 2 chromatids together to form a chromosome. So they say here it is going to split. So when it splits, chromatids are going to do what? To move to the opposite sides or to the opposite poles. So once you see that happening, you see now single things, chromatids. We call them chromatids because they were joined together by a centromere. So when they are pulled now to the opposite poles, now you know that this must be anaphase 2 because I'm not seeing a whole chromosome being moved to the opposite pole. I'm only seeing 1 and 1 being pulled apart. Right. So that is how you identify this stage. Then the last stage will be telophase. Telophase 1 and telophase 2. What is happening during telophase 1? The nuclear membrane reforms. So once you start seeing this new formation of the nuclear membrane, then you know that this is telophase 1. The cell membrane constricts. So here, the cell membrane constricts and forms what we call a cleavage here. So this is the constriction of the cell membrane. You see it's coming this way, coming that way. That is the constriction. Cytokinesis occur. The separation or the division of the cytoplasm. Remember, this is the cytoplasm of the cell. It's going to split. So you know that all of these tell me that this is telophase 1. And then in telophase 2, you must see 4 things or 4 cells sort of that are forming. Now, each daughter cell has half the number of chromosomes. So you're going to see in here you have 1, 2, 1, 2, 1, 2, 1, 2. And whereas this cell started with 4 homologous pairs. I hope you remember. 2 homologous pairs. So meaning there were 4 chromosomes. Now you see every cell now has half of where you started from. Then 4 haploid daughter cells are formed and they are genetically different. Why are they genetically different? Because of crossing over that happened at prophase 1 and also because of the random arrangement in metaphase. So I hope you remember these stages. Then also you will see that cleavage is going to form here. The cytoplasm is going to split, but you must see 2 things splitting into 4. Whereas in telophase 1, you see 1 splitting into 2. Right. Then those are your phases. So what is nondisjunction? We touched on nondisjunction and it has its consequences. The spindle fibers, they must separate. They must pull apart. But sometimes these spindle fibers, as they attach to the centromere, they fail to separate. And when they fail to separate during meiosis, what's going to happen? Members of one pair of homologous chromosomes, they fail to become separated. So when things were like this and they were supposed to be separated like this is the failure to separate during anaphase 1, and may happen in anaphase 2 also. So in anaphase 1, it will be chromosomes not separating. In anaphase 2, it will be chromatids failing to separate. Now, if there is non-disjunction of chromosomes on pair 21, so if on pair 21 the chromosomes fail to separate in humans, it leads to a formation of an abnormal gamete. So, what is an abnormal gamete? That gamete is going to have an extra copy of chromosome 21, because remember, chromosome 21 are supposed to separate, but in one gamete it's going to come with 2 chromosomes that did not fail to separate. Now there is an extra copy, and there is supposed to be 1 chromosome from the father, 1 chromosome from the mother, but there will be this extra chromosome at chromosome pair 21, and we call this trisomy 21. But it can lead to meiosis. No, it can lead to Down syndrome if this happens at chromosome 21. Now, 2 extra gametes are going to receive an extra copy of the affected chromosomes. Then 2 gametes are going to lack that chromosome. You see this part here? I don't want you to focus on it because it leads you to answer the question in a negative form. When you're asked to explain what are the consequences of non-disjunction at chromosome 21, you will focus more on telling us about the chromosome that did not receive, that will be empty, that will be what? No. For Down syndrome, focus on the extra copy on chromosome 21, not the one that has less, because the one that has less, it's another syndrome, but it is not Down syndrome. So for Down syndrome, focus on the extra copy. So your story, when you are explaining it, focus on the extra copy, not on the one that does not have, or the less. Then you'll be explaining Down syndrome. Right, those are the things that you need to look out for. So this is just a diagram to explain to you with the pictures that I was drawing there, but this is a clear one that shows you how non-disjunction can occur. So if I draw a line here, okay, let me draw a line. You see that dotted line that I'm drawing? I want you to see between the 2 sides. The left-hand side, on my left-hand side, you see the normal separation, and on the right-hand side, you see non-disjunction. So, under normal separation, you would expect that these homologous pairs will separate like this, isn't it? You are going to hope that each chromosome is going to come into each gamete during meiosis 1, because you are talking about homologous chromosomes. During anaphase 1, because anaphase, that is where they separate. So you expect them to be like this. And then, unfortunately, under non-disjunction, they fail to separate. So they fail to separate. They fail to separate. They both come into this cell over here, and it leaves this one with nothing. And remember, we said for Down syndrome, don't focus on the one that has nothing. Focus on the one that has an extra copy. So for non-disjunction will be happening in this way. Then further, during the second meiotic division, these chromosomes, you expect them to further divide like this. 1 chromatid into 1 gamete, 1 chromatid into 1 gamete. However, when they fail to separate during the second meiotic division, they might both come into one cell. So it means this cell has an extra copy, meaning this chromosome or a gamete of 24 is going to match with a gamete of 23, and we don't have 46. You see, there is an extra copy here. Now the other one will be left with nothing. But here, under normal circumstances, normal, remember, they will come like this, each chromatid represented in the cell, and then this cell, because it had nothing, it cannot further divide into anything because there was nothing to begin with. So I hope this picture just shows you or summarizes to you what Nondisjunction is. And then the differences between mitosis and meiosis. Mitosis occurs in body cells, and meiosis occurs in sex organs, your testes and your ovaries. Then both karyokinesis and cytokinesis occurs once in mitosis. But in meiosis, they happen twice. When? During meiosis 1 and during meiosis 2. Then 2 daughter cells are formed during mitosis, but during meiosis 4 daughter cells are formed. And the daughter cells in mitosis are genetically identical to one another and to the parent cell. So everything is the same in mitosis. So if you started with 8, the daughter cells will have 8 and the mother will have 8, the mother cell will have 8. But with meiosis, the daughter cells are genetically different because of variation from each other and even from the parent. Then the chromosome number remains constant, 8, 8, 8, 8, 8. But here, the chromosome number is halved. Remember, we're taking it from a diploid situation to a haploid situation with meiosis. Right. So that is your topic, meiosis. Then we come to the next topic, which is genetics and inheritance. Remember, we said this is quite a big topic. It is going to weigh 48 marks out of your 150. So you need to go and revise thoroughly with this one. So you see there are so many subtopics you need to know on genetics and inheritance. So let us go into it. So you need to know all the concepts. Terminology is very important. Then your monohybrid crosses and your dihybrid crosses, your sex-linked inheritance. Which ones are sex-linked? Because most of the time, when you are given a genetic cross to solve, you use X and Y unnecessarily without reading that this condition that I'm dealing with, is it sex-linked or is it just a condition, a normal condition that does not affect the sex chromosomes, which are the gonosomes, your X and your Y. So you must check that, the sex-linked inheritance. Then the pedigree diagrams, be able to interpret and trace. Then genetic engineering and paternity testing. Most of the questions for genetic engineering will come from a case study or from an extract or something like that with biotechnology and all that. So remember stem cell research and your GMOs and cloning. Right. So what is this terminology that we were talking about? Because genetics has intense terminology, some of this terminology you use it interchangeably or you find yourself having confused it. Check. A chromatin. Think in your mind. Chromatin. Which one is that one? Chromosome. We've just spoken about the chromosomes. So the chromosomes and a chromatin, not the same. So when you want to write chromosome, please write chromosome in full. Because if you write chromatin and it is not relevant for that particular answer, you are going to lose marks. Then, this one, we see it a lot. Genes and alleles. Someone wants to say, no, the alleles of the parent or the alleles are giving this particular character, and they will say genes. And when you say a gene, still a general thing. So you need to be specific. Know when to use the term gene and when to use the term allele. Then the dominant allele and the recessive allele. No, when we say an allele is dominant, what do we mean? When we say an allele is recessive, remember the recessive is the one that is going to be masked by the dominant one. The dominant one is the one that is going to show in the offspring. in a heterozygous situation. Then a phenotype, your physical aspect, your physical appearance of an organism, your genotype, the genetic makeup of that particular organism. Homozygous, having the same alleles or of the same size and dominance. Then heterozygous, different. So if you can just use the letters H's, homozygous can be this or it can be this. So you see they're the same. With heterozygous, it will be a big H and a small h. Hetero, not the same. You see? Right. So please make sure that you learn when to use these concepts as you are answering those previous question papers that you are studying with. And then remember the grandfather of genetics, Gregor Mendel. He has his laws that govern the principles of genetics, what is happening. So you must know Mendel's first law of inheritance. This is the law of segregation. Remember, he was working with the peas and he started segregating, and then he saw that, so now the law of segregation. Then the second law of inheritance is the law of dominance. So you must just remember, law of segregation, law of dominance. Then Mendel's third law of inheritance are the law or the principles of independent assortment. You cannot forget those. Segregation, law of dominance, and independent or the law of independent assortment. Those are the laws that you need to know that Gregor Mendel found in his research. Then, how do you approach a genetic cross? Most of you are also losing marks here. Free marks. So there is a way. So let us check first at the terminology that you will come across here. The visible trait, that is the phenotype. Example, you must look at the visible trait. When you are given a problem, check what is the visible trait they are talking about. Something that you see on the outside or the physical aspect, like tallness, shortness. This is something that you can see in a plant. Then the genetic makeup of an individual, it is the genotype. So the genotype will be where now you use the letters. TT, that is homozygous, heterozygous TT, and recessive TT, that is homozygous. Right, so check the genotype. What is the genotype in the question of whatever that you are given? Then it is the genetic makeup. It's not something that you can see on the outside, but you can work it out using a Punnett square. Then the alleles segregate or they separate during meiosis to form gametes. So during the process of meiosis, we just saw that gametes are formed. So the alleles are going to separate, meaning the alleles for the mother and the alleles for the mother and for the father will come separately. And then in an organism, that is where you're going to see whether the mother gave a dominant or a recessive one, and then it becomes a heterozygous or a homozygous situation. It is because of the segregation that took place during meiosis, because we were halving everything. Right. Then, during fertilization, the individual is going to get one allele from each parent. So this is what we are talking about. Each gamete will be carrying just one allele because they were segregated. Then, if they are carrying a dominant allele, let's say this is a sperm cell, and then coming from the father, and then there is another ovum. Let's say this is an ovum coming with a small letter T, coming from the mother. Right, so during fertilization, these alleles are going to do what? An individual is going to get one from each parent. Then this offspring from this fertilization is going to have big T and small t. So this is what is meant by this statement above here. So getting one from each parent. So that is how inheritance works according to Mendel. Now I want us to focus on where to collect the marks on a genetic cross. Here also, I need you to be able to at least get 3 marks. You can be asked this question 3 out of 6. But the 3 marks are a given. They are for free. Do not lose them. Even if you don't know the answers that come here, even if you don't know, please make sure that you write the format correctly so you can obtain marks. So let us check where can you obtain these marks for free. Today I'm giving marks for free. Remember that by writing P1 and F1 and meiosis and fertilization at the correct places, you are going to get 2 marks. Let's check. You start your format by saying whatever. This is my first parent generation. Parent generation gives me the F1 generation. Then you write your P1 up above there and your F1 right here. You already obtain 1 mark for writing them there at the correct positions and at the correct sequence. You can't start with F1 and then P1 because some of you in the exam, when you've forgotten, you write F first and then P first. You start with the last letter. P1, parents, P, parents. Parents generation P1, then giving you F1, first filial generation of the offspring. Then, meiosis and fertilization. So you must just remember, meiosis comes on top, then in between is gametes, then fertilization. So once meiosis and fertilization are at the correct place, that is another mark. You have not yet solved the problem, or you have not yet solved the question that you are given, but already you have these 2 free marks. The first free mark that you can get is this one here. The phenotype. Why do I say you can get this mark for free? From the question, you will be given a statement to say if a mother that is black is crossed with a father that is white. Already you have the phenotypes of the mother and the father. They are telling you their physical aspect. White, black. Then you write phenotype, first one, black mother crossed with white father. You see, already you get that mark from the question. That is for free. Then you have my 3 bonus marks. Do not lose those marks. Collect those marks. And I don't want to hear anything. It's non-negotiable. Right. Then the genotypes. You can be given the letters to work with in the question. Then you start figuring out whether they say this black mother is homozygous or is heterozygous for black. Then you will write the correct letters here and the letters of the father, and then you separate them. Separate the letters. Here you can even use the circles and not write this. Just put the circles to show that segregation. Now these are gametes. Or you can just write them with spaces apart to show that they have been separated. And then those gametes, now you put them into fertilization. You're going to put these ones here and then these ones here on top. Then you start crossing here. And once you start crossing, you will tell us the phenotype to say 1/4 of these offspring are black, or 1 is 1 black, 3 are white, or 3 are brown, depending on the situation. And then what are the genotypes that you found? And the question can further ask you for a percentage or a ratio. And if that is the case, you will see that this will carry a compulsory mark. So if there is a compulsory mark to be collected, the marker will start by looking for this mark. So make sure that you get it. And then, okay, this is just to show you that you gave marks there for P1 and F1 and also for meiosis and fertilization. So this is how you are going to answer your genetic cross. Then types of inheritance. When you were doing all the crosses, you checked there are monohybrid crosses, complete dominance, incomplete dominance, co-dominance, sex-linked, and dihybrid. Now, what is a complete dominance? Yeah, I'm interested on these ones to check. Do you know? When do we say this is a complete dominance? When do we say it is an incomplete dominance? And when do we say it is a co dominance. You must be able to identify that in the question that you are given. Check if they tell you that all their offspring are like 1, 2, 3, 1, 2, 3. Sometimes they don't tell you, they want you to work it out and then you will tell them. So it can happen vice versa. Let's check. The first one, complete dominance. So if you check this true breeding green pot, if it is crossed, you see this is a true breeding green pot which is crossed by a true breeding yellow pot. So yellow crossed with green. But you see now the green is represented by the big G's and the yellow is represented by the small g's. And remember, you use the same letter. You can't say green is G and then yellow is Y. If you chose one letter, you must stick to that letter for the other, the genotype of the other of the opposite allele. So please make sure that you don't differentiate the letters and say G and Y. Right. So small g represents yellow, big G represents green. But then you see when they cross them, remember these are gametes. When they cross them, all the offspring are green. So what is a complete dominance? When all offspring have the characteristic of the dominant allele. So that is a complete dominance. So only one parent's phenotype is evident in the offspring. So that is that parent, meaning is dominant over the other because their phenotype is shown in the offspring. So we know that this is a complete dominance. Then let us check the other type of inheritance, incomplete dominance. When you cross a white flower with a red flower, and do you see that they used the different letters here, and you use capital letters for incomplete dominance and co-dominance, right? But you need to identify from the answer that which one is which. So read your questions carefully so you can identify the type of dominance that is shown there, right? So how do you know that this is incomplete dominance? Look at the offspring. All the offspring, what are the colors of the offspring? They are pink. Did we cross a pink parent with a white parent, or what? Did the parents look anything like pink? No. So neither of the parents' phenotype is evident in the offspring. So the offspring brings a new color on its own because both the phenotype of the parents are in complete dominance. No other alleles are dominant over the other. So they are expressed in a way that they form an intermediate color. So this is an intermediate color that will be pink because we did not cross pink, we crossed white and red, it gave us pink. That's how you know that this is incomplete dominance. You must look for that intermediate color that you do not see in the parents. Right. Then the last of types of inheritance is co-dominance. Co. Sometimes this word co can be used for company, co, together. Right. So co-dominance. Let us see. Both of the parents' phenotypes are evident in the offspring. So when you cross a white cow with a red cow, with cows, you don't call it a red and white cow, ne? It gives a roan color. So this is the color. So you see here, you see white. You also see red. What does this mean? Both of them are co, meaning they both are evident equally in the offspring. So the offspring will have both colors. They do not mix. Both of them appear as they do in the parent. So this is an example of co-dominance. So I hope you remember your types of inheritances. And then sex determination. This is a karyotype that is of a male. So this is a male. When you see this sign, You must know. This is a male. Then all of these chromosomes from number 1, we can match them. Chromosome pairs. These are pairs because this is a complete human being. So they are pairs, meaning they appear from the mother and the father. In each column, mother, father, mother, father. But they are in sets. Set number 1, number 2, 3, 4, going on and on and on up to number 23. So all of these chromosome sets are called autosomes. Autosomes is number 1 to 22. So they are called autosomes. These are ordinary chromosomes that can carry, or they have genetic material for different things. However, when you get to chromosome number 23 in a karyotype, that is your gonosomes or your sex chromosomes. So what are the sex chromosomes? They are the ones that tells you whether this person is a male or a female. And this one is of a male because it is XY. Remember, XY. And X is a little bit bigger or longer than the Y. So the Y is a shorter chromosome. So that is why you will be able to tell that this one is the karyotype of a male, because at number 23, these chromosomes are not equal in shape, because if they were equal in shape, then it will be of a female. Let me show you a karyotype of a female. Now this one is of a female. And when you take a look at this chromosome number 23, you see now it is XX. Do you see now XX are equal? So this is how you are able to tell that this is a female. Then sex determination. This is it is a given that you must know. It is a fact that every time when you cross a male or a mother with a father, there is a 50-50% or a 50-50 chance of getting a male or a female child. So when you work it out, a genetic cross, there is always a 50% chance of getting that. So it will be your female and your male. When you work it out genetically, you want to find out. Right, okay, we're not going to put that one here. Okay, let's just put X here. Yes. So there will be nothing here. Here is just gametes. I'll write G. So here it will be your XY. Here it will be your XY. Here it will be your XX. This one and this one will be your XX. So every time there is a 50% chance of getting female kids and 50% of getting male offspring. So it is going to be 2:2 or 50-50. So you see, always with sex determination, these are the chances, right? And the males are the one that determines the sex of a child, because they can give the Y chromosomes, whereas the females can only give the X chromosomes. They do not have a Y chromosome to give. Then the sex will be determined by the male. Then sex-linked disorders. This is where I want you to be very, very careful. Genes located on a sex chromosomes are called sex-linked genes. So on your X and your Y, sometimes there are genes that are there that may be affected by certain diseases. So we'll say those are sex-linked. Why are they sex-linked? Because they are found on the sex chromosomes, on the X and the Y that I've just shown you. Now, in humans, the term usually refers to X-linked characteristics, or the genes located only on the X chromosomes. Fathers can pass the X-linked alleles to their daughters, but not sons. Why? Because they will be on the X chromosomes, and fathers only have one X and one Y. If a father gave a son, then a son will only inherit a Y from the father and not the X. So that is why we say they can only pass the X-linked alleles to their daughters if the father gave the X chromosome. But mothers can pass the sex-linked alleles to both sons and daughters, because mothers can give an X, it can be a daughter, can give another X, it can be a son. So mothers only have X's to give. So if she gives the affected X, it will mean the son or the daughter can have that particular sex-linked disease. And please don't just use the X and the Y's if you are not told that it is a sex-linked disease. Use your normal letters. Only in cases where you are told that this particular disorder is caused or is found on the X chromosome or on the Y chromosome, that is the only time you will use your X chromosomes or your Y chromosomes. So you do not use this if you are not told, or unless you are talking about hemophilia and blindness, which are the ones that you studied for this year. So this is just a diagram to show you that mothers can pass to their daughters and their sons, and the fathers can only pass to their daughters, not their son. Right, so this is just a diagram that was going to show you that. And then color blindness is one of the X-linked diseases that you studied. So there are people who cannot see the red-green color, or they do not have the alleles for that or the genes for that. Then their rods and cones, they just have that confusion. A normal person with normal rods and cones or cones will be able to see this is a green cap, this is a yellow t-shirt, and these are red sneakers. But a person with a red-green confusion color blindness is going to see all these things when you put them together as yellowish curry, and it is a genetic disorder, and it is carried on the sex link. So the example can be a normal female. Okay, let us take a look at this diagram. This female will be normal with XX, and then this male will be normal with this green one normal. But the ones with, okay, this woman is a carrier of color blindness. So you see, the allele for color blindness is this red one, but the blue one is normal. So this male is normal. So this female can give birth to a daughter that is colorblind, or normal female who carries the normal female who carries this color blindness allele, because she has another X to mask this one. And then she can also give birth to a male who is colorblind. So if this lands on a boy child, then the boy child can end up being colorblind. So this is just what. And then another set of genetics groupings that you need to remember are blood groups. Also here, we have seen you using the eyes unnecessarily. So check. If they are talking about blood groups, this is the only time that you are going to use the alleles for blood groups. Remember, we only have 4 blood groups and they represent multiple alleles. So you have blood group A, B, AB, and O. Then what are the alleles if you were to cross the blood groups? For blood group A, you can have a homozygous blood group A or a heterozygous blood group A. So you represent that with a big I or capital I and then capital A, or with a capital IA and a small I, which shows the recessive allele that is going to be masked by the dominant allele of blood group A. You can do the same for blood group B. Then for blood group AB, this shows what? Right, I want to show you now. This is complete dominance because it is completely dominant over this one. And then for blood group B, also complete dominant because it is blood group B over B. Then with blood group AB, very interesting, both AB are represented in the blood group of this offspring. So what type of dominance will that be? That is going to be co-dominance. Remember, both of them they are a company, they are together. And then blood group O is, you can represent with recessive eyes. So these are small letter I's. They must be in a homologous state. So this just shows complete dominance. It cannot appear in any other way. So you use those blood groups. And then genetic crosses, genetic pedigrees or genetic lineages. This is a genetic lineages. Remember, pedigree diagram, you are going to be asked, what type of diagram is this? This is a pedigree diagram or a genetic lineage diagram or a genetic pedigree. Now, a genetic pedigree traces the inheritance of characteristics over many generations. So here, the trick is for you to know how to solve it. Check your key all the time. Check your key. You will be given a key, but at some stages you might not even be given a key, but you must assume, based on the information that you'll be given, work it out that, okay, here you are told that the circles are the females, but the circles that are empty, that are not shaded, are non-hemophilic females, and the circles that are shaded are hemophilic females. So already on your diagrams, you can check, are there any females that are hemophilic? No, because you don't see any females that are shaded black. Then you see the squares. The squares, you have the empty, non-shaded squares. They are non-hemophilic. And then the shaded squares represent hemophilic males. Then you can already go to your diagram and check which ones are hemophilic males. Then already you know these are males and they are hemophilic. Then you can work out the genotypes based on what you are given of these particular people, and also check where did they get it from, because you trace it backwards. You don't start filling it from the top. You start from the bottom up, because then you are tracing the inheritance. So for you to trace inheritance, you must go back in time. Right. So you must know the meaning of the symbols and the lines that are used in a pedigree. So if you see in the first row, you will see a horizontal line. You know that it represents a marriage line. Then you see it going down. It represents the descent. Now, when after the descent, you see a line going across, you know that those people under that same line are siblings. So this one and this one are siblings. Let me just represent them with stars. Then you know that they are siblings from these parents. Then with the parents, you need to work or check the key to say which one is the female. Then the circles will be the female, then the squares will be the male, and then you also check what are they affected with, or affected or not affected. And then the first row will represent the generation. So number 1 here will represent the first generation. The second row will represent the second generation going forward. So this is how you interpret your genetic pedigree diagram. So how do you work it out? First, you must study the key. One thing that we said you must be very important. Now, look for the dominant and the recessive characteristics. How are you going to see that? Check the inheritance. Check where these people are inheriting. Now, fill in the genotypes of the individuals with the recessive condition. Go crazy. Go to all the ones that you see that they have the recessive conditions, small letters. Fill all the small letters from down upwards. Now, each individual with the recessive condition inherited one allele from each parent. So now you know, for an offspring to have both of these, it means one must have come from each parent. So your parents, you can work them out to say this parent must have had an A and this one must have had an A. So for this offspring to have both the A's like this, already you can fill that information in about the parents. Then work backwards and fill in one recessive allele for each parent, because you've worked out all the recessive alleles. Now, if the parent has the dominant characteristics, fill in a capital letter as the second letter. So where are you going to get this information? You are going to get it from the key or from the information that you are given. Then any other individual showing the dominant characteristics is most likely homozygous dominant. So with the 2 capitals, then you will just end up filling all of this, and that is how you are going to work out the genotypes of all the members, the genotypes together with the phenotype of all the members of this family going back in generations. Right. Then you can use genetics to prove paternity testing. Paternity testing, you have blood groups. Remember we said we only have 4, AB, and O. Then how can you use these blood groups to prove paternity? Remember, there can only be 4. So this cannot be a very valid and credible way to prove paternity, because people can either have blood group A, blood group B, blood group AB, or blood group O. So if you want to say the father next door is the father to the child of the mother in the front opposite, and you find that both that father and that child, they are blood group O, how can you be certain that it is that father who gave that child the allele for blood group O? You cannot be certain because there are so many people with blood group O. However, with DNA profiles, you can certainly say that these bands match the bands of a child. Therefore, this child, because you know you are working with genetics, this child's nucleotides or the arrangements of the nucleotides are familiar or are they are similar to this particular father, then that makes them the father. You can say that with conclusion. But with blood groups, it cannot be conclusive. Then the last topic that you should look at, this is just an overview of the topic. Please make sure that you go and look at this topic, that is evolution. Check the evidence for evolution to start with. What are the sources of variation? We've spoken about them when we were doing meiosis, so you get a mark for that. And then check these evidences. Yes, we've spoken about evidence. Check artificial selection and evolution. Okay, the ideas, these are very important as they appear. Lamarck, what did he say? How did the organisms evolve? Was his theory accepted or was it rejected, and why? Then Charles Darwin, how does he say evolution took place? Then punctuated equilibrium by Eldridge and Gould. What did they say happens during punctuated equilibrium? Then artificial selection, then evolution in present day, that is where you are talking about your COVID-19 and all of that, the evolution in small microorganisms. Then be able to identify or describe speciation. How are species formed? Remember the geographical barrier. You need to identify the geographical barrier in terms of the information that you are given until you come to species. And when you are describing speciation, do not forget that you start, everything is population. Then at the end, you will only refer to species, because you cannot be forming species and talk about species. You must say population. Then also know the reproductive isolation mechanism. So once these organisms have speciated or they become new species, why or what keeps them apart or what keeps them from reproducing? So those are species-specific behavior or courtship behavior, breeding at different times, prevention of fertilization, adaptation to different pollinators, and infertile offspring. So these are some of the reasons that you can look out for. And then you must look at human evolution. With human evolution, you will be looking at your scientists and those characteristics that you share with the African apes, what makes them different, also the differences. Remember your skulls, your dental, your hip bones, and also remember your scientist and out of Africa hypothesis. What are the reasons? Why do we say this is out of Africa? And then the fossils to prove that, and also evidence for human evolution. Please make sure that you go through all of that with the worksheets that you have. They will enable you to go through this and use additional material that you have to go through this. And good luck for your exam tomorrow. And thank you so much for watching this lesson. I'll see you again.